Okay, so my presentation is essentially a package of kind of the neuropharmacological mechanisms of ketamine squeezed into 20 minutes, and I hope that will be kind of an instructive lesson for those who are not maybe so familiar with ketamine, but also I tried to incorporate aspects that would hopefully be useful for also those who are working in the field, and kind of present also a little bit of the kind of latest ideas related to ketamine's pharmacological mechanisms. Here I have the title Neurobiological Mechanisms Underlying Ketamine's Antidepressant Effects, and I chose neurobiology as kind of the mechanistic insight term here, because I think that's probably closer to what ketamine ultimately is doing. So at least to me, it appears that ketamine is essentially taking use of many of the neurobiological mechanisms of the brain in order to bring its antidepressant effects. And while I'll be focusing here mostly on pharmacology, I hope maybe the next speakers will then give more insights maybe into the clinical use and also the kind of psychological aspects of ketamine. And finally, before we get started, here just an image of a pyramidal neuron and the branching dendritic trees. And here on the left, you have a picture of another kind of tree covered in snow. This is how I believe it more or less looks like in Finland at the moment. And to be honest, I would really love to be there and enjoy the snow, but unfortunately there has been no snow in New York since December. So I'll have to wait for probably some time until the end of this year. But anyways, let's get started. Just a few words about depression before we go into the kind of molecular mechanisms. Obviously, depression affects a huge amount of people around the world. It's a leading cause of disability, and it's a major risk factor for suicide. Depression causes immense individual suffering to the patients themselves and also to relatives, and of course also immense economical burden to society. So there's many reasons why we would want to treat depression as effectively as possible. The current treatments are typically traditional antidepressant drugs like SSRIs, and in the best case scenario, that's combined with psychotherapy. Unfortunately, quite limited due to the resources. Unfortunately, the commonly used treatments, or the drugs in particular, work slowly, if at all. And I think it's clear that there is a need for better treatment options for depression, and not only medications, but also other kinds of treatment options, physiological treatment options, perhaps. So these traditional drugs, SSRIs, are drugs that are taken typically every day for prolonged periods of time for the patients to then see any amelioration of the depressive symptoms. Typically, it may be that a patient has to go through several different drugs, trying out kind of different classes within the traditional drug categories. It may take months, even years, to find a drug that is finally useful. And even when it's useful, it may not be helping that much. And there's also a large amount of patients that do not really respond to the traditional drugs. And this is where ketamine and other kind of special treatments like ECT come in, and those are particularly one of the few options available for severe treatment-resistant depression. And ketamine, when given in a clinical setting, is quite different from these traditional drugs. Typically, it's given as an intravenous infusion, 40-minute infusion, sub-anesthetic dose, 0.5 milligrams per kilogram is kind of a standard these days. And the really rapid effect is by this information, right? So maybe during the infusion or immediately after that, getting the benefit from the drug and responding with the reduction of depressive symptoms. And indeed, these ketamine are very rapid. They're also sustained. So they typically are the strongest during the following day, at least according to many clinical trials. And then they may stay. Now, the depressive symptoms may stay away or stay reduced for a week or two, maybe. And unfortunately, after that time period, approximately, these symptoms then typically start to re-emerge. So the re-emergence is actually quite gradual. It's not like a week goes by and then the symptoms come back, but they slowly start to then reappear, and then new doses of ketamine are typically needed to be given. And this is, of course, not maybe the case for all patients, but it's kind of the general picture. And still with ketamine, there's also a number of patients that still don't benefit from even ketamine treatment. So I think understanding why the ketamine's effects then fade away and why not all patients benefit would allow us to maybe optimize the treatments or maybe develop new ways how to tap into this potential of ketamine for the treatment of all patients. So the main question of this presentation will be how ketamine works, and we'll be tackling that by going straight into the brain. Here you can see an image of two neurons, one neuron here sending an action to another neuron, with the dendrites being here of the second neuron, and there's a place here rounded by this square or this ring, where the neurons contact, and there is essentially what is called a synapse. And here you can see the presynaptic side of a synapse and the postsynaptic side of the synapse. And the idea with these synapses is that they are points where the neurons can release chemical neurotransmitters to the synaptic cleft here in between, which then bind to the postsynaptic side and can activate the subsequent neuron or the postsynaptic site. And if we particularly focus on the cellular membrane here on the postsynaptic side, it may be something which looks a little bit like this. And here you have the cellular membrane or the phospholipid membrane, and you have a receptor protein sitting here on the membrane. This is probably not exactly to scale, but it's more or less probably what this could look like. Anyways, this here is the N-methyl D-aspartate receptor, or NMD receptor, which is the kind of the main target of ketamine for its effects to then appear. It's also a primary or acts as a target for the glutamate, which is a primary ligand of this receptor. And this receptor functions as an ion channel, which means that ions can pass through the channel pore to then have various effects on the neuron here. And these channels are quite important in learning, memory, plasticity, kind of synaptic homeostasis, and the modulation of synaptic strength in synapses. And also these properties in, for example, the formation of memory traces are important when high doses of ketamine are given, which typically kind of shut down the brain more or less. And with very high doses, there's typically also amnesia, and that's basically a result of the properties of ketamine kind of making the memory formation system not functional. So I can replace the receptor here with maybe a little bit simplified version for just the purposes of showing how it works. And here we have the ketamine molecule on the top. So this is kind of actually ketamine as such contains two isomers or stereoisomers, which are like the left-handed and the right-handed forms of ketamine. So they're quite similar. They're both hands, but if you try to superimpose one hand over the other hand, you cannot essentially make your left hand into your right hand. So they have a little bit different properties, but they're essentially both can do things rather similarly. Here, the S-ketamine is particularly more potent at blocking the NMDA receptors and is typically used more in anesthesia. For ketamine to act, it must have glutamate bound to the receptor as well as its co-agonist glycine, which then opens the ion channel pore here and allows ketamine to bind in and block it, and essentially blocking ions from entering the channel, like calcium here. And this then mediates a kind of lot of the basic level mechanism of ketamine. So this is a little bit simplified version. There's also many other things related, but let's not go to these things within this 20-minute time frame. Now, in order to understand how subanesthetic doses of ketamine particularly work in the treatment of depression, we have to look at kind of a bigger picture. And here is again another synapse, and there's various spots where there are these NMDA receptors on this excitatory synapse, but also on this inhibitory interneuron here on the left right corner. And what subanesthetic doses of ketamine have been particularly thought to do is blocking the NMDA receptors at these inhibitory interneurons here on the left. So instead of maybe having these primary effects come through the receptors on the postsynaptic sites, it would be actually blocking these inhibitory interneurons here. And by blocking their activity, ketamine is essentially taking away kind of the brake system of the brain. And by taking out the brakes, you essentially result in the disinhibition of the activity of this excitatory neuron or synapse, and thus increase then the release of glutamate from the synaptic membrane to the synaptic space here, and then it can bind to receptors such as AMPA receptors, which are similar ion channels as NMDA receptors, also binding glutamate and activated by glutamate. And they can then pass through ions and essentially result in changes of the voltage and the depolarization of the postsynaptic neuron, and also the activation of neurotrophic mechanisms, which then reside inside the neuron here. So I have drawn these inside the spine here, but typically many of these mechanisms, like these transcription events, take place in the nucleus almost completely. What ketamine is thought to do by increasing the activity of the synaptic contact and activating the AMPA receptors is that it then results in the rapid translation and release of brain-derived neurotrophic factor, BDNF, which is a key player in synaptic plasticity and strength, which then activates different receptors and molecular pathways, ultimately resulting in changes in protein synthesis, changes in gene expression, and ultimately facilitating the synaptic plasticity and synaptic strength of a particular contact here.
Now, ketamine is also thought to increase synaptogenesis, which essentially means the formation of new synaptic contacts. So here, for example, you can see a pyramidal neuron, and if you look at one of the dendritic branches, one of the apical dendritic branches here, you can see these small spines protruding or protrusions here from the dendrite. And what has been shown in animal experiments is that during baseline conditions, when you can do quite precise imaging of these dendritic spines in live rodents, and you can find a certain number of spines, and after having the animal subjected to chronic stress conditions or chronic corticosteroid treatment, a stress hormone, some of these spines here marked with the red arrow will then disappear. So there's like some sort of an atrophy of some of these cortical spines. When these animals are treated with ketamine, and 24 hours post-ketamine in particular, some of these spines can be recovered. So those are marked here with blue, suggesting that ketamine is able to restore not only functional processes, but also structural processes, at least in terms of the synaptic spines or the dendritic spines here. Now, if you look at the kind of the bigger picture in terms of more global effects of ketamine at the level of networks over the period of ketamine's kind of effects from the immediate within hours effects to those happening during the same day, and then to those happening during the following days, I've shared this slide into these kind of 3 boxes to just kind of group those effects a little bit. So here on the left, there is the start of the ketamine treatment, and on the right, there's the maximum response. Typically, this takes place 24 hours after the treatment. So what happens immediately within hours of ketamine treatment? The most typical finding here is the increase of cortical activation, particularly the prefrontal cortex. So this is the brain area which kind of acts as the orchestrator of our kind of thoughts and actions, if you can say it so. And what ketamine seems to be doing, as shown by the previous molecular evidence, also by imaging methods, it has been shown to increase the metabolic activity of the prefrontal cortex, increase glutamate neurotransmission, and then increase high-frequency gamma oscillations. So in terms of kind of functional magnetic resonance imaging, one of the features that have been detected already to happen within hours is the increase of the prefrontal cortex global brain connectivity, essentially how the cortex is then connecting to other brain regions and how the different regions are talking with each other. Now here, I put a question mark because, to be honest, there's also studies which are saying that this is not the case. They haven't seen this kind of an effect with ketamine, or they've seen completely something opposite. So this is a problem with many of the fMRI studies, that there is, at the moment, I think there's still, the method hasn't been developed that far that we could really for certain say which regions and which findings are completely kind of indicative of ketamine's effects. But they, at least I think, combined with these other measures and also combined with the rodent data here on the lower part of this picture, where the normalization of this kind of prefrontal cortical ensemble activity has been shown, and also this neurotrophic pathways activating, I think it's reasonable to assume that there are changes in connectivity taking place quite rapidly. And since there's also the kind of rapid amelioration of the depressive symptoms, I would expect that this somehow also seen on the level of these brain networks. So what about kind of effects during the same day after maybe 4 to 8 hours or so? One of the key findings related to kind of depression and traditional antidepressants in general has been that of the kind of normalization of the activity of the subgenual anterior cingulate cortex. And this area is located basically here. This is the anterior cingulate, and the subgenual anterior cingulate is particularly this downmost part here, which is under the genu or the knee of the corpus callosum. And this is particularly maybe this kind of the most ventral part has been thought to be connected with kind of emotional processing. And the anterior cingulate in general is kind of related in cognitive flexibility and kind of reward processing and many of the similar higher order cognitive functions as the prefrontal cortex as well. And in particular, what's been found out related to the subgenual anterior cingulate is that the activity patterns and connectivity patterns of this area correlate pretty well with the severity of depressive symptoms and with, for example, depressive rumination. And as traditional antidepressants take effect, typically the activity is normalized also in this area. And there's also been some studies where there's been deep brain stimulation of particularly the Brodmann area 25, which essentially is within the subgenual anterior cingulate region. And the deep brain stimulation of this area is also helpful in treating depressive symptoms. So these findings pretty much suggest that this could also be a very kind of crucial target for ketamine's effects. But here again, there are also findings that are much more unclear or point to the other direction. In terms of other things, here MEG studies have shown that responses to somatosensory stimuli are enhanced in ketamine responders, which kind of suggests to me at least that these acute changes of ketamine ultimately result in, for example, the strengthening of synapses, which is then seen in these responses at these further time points. And in general, the normalization of connectivity and metabolic activity takes place during these time slots. And just as an example, I've here also added a couple of things related to the default mode network. So this is also one of the kind of key functional networks associated with depressive symptoms and rumination, for example. And it composes of many different structural brain areas and regions. And in particular, the default mode network is kind of thought to be a network which is active during resting, when the brain is kind of not actively engaged in particular task activity, if you can say it so. And it's kind of thought to be activated in kind of spontaneous thinking and kind of self-related feelings and also like depressive rumination. And I just have here one study which shows that the normalization of connectivity between the insula and the default mode network 2 days post-ketamine has been shown to take place. And here the insula is also another brain region thought to be kind of a key area in the integration of kind of this kind of external emotional stimuli and also kind of the switching between other networks and the default mode network, which could be important for also the emergence of depressive behavior and symptoms. And what's interesting to me here is that these connectivity changes appear to return to baseline 10 days after the treatment, which is kind of in line with the time schedule of ketamine's effects, that the symptoms typically then reappear at one point. And somewhere along this 4 to maybe towards the 8 hours to 24 hours, there's also a restoration of dendritic spines in these animal studies. So this suggests that the dendritic spines are probably something that is then more related to how the effects of ketamine are then sustained. And this is also the message that's been coming from the animal studies, essentially. So this is just a very quick overview of some of the findings. There's many other findings and many other key regions that have been reported, but unfortunately I won't have time to go into more detail here. But what I will spend the last few slides and the kind of try to end this presentation with is to present some of the ideas I've been working with myself in the previous couple of years, trying to understand the answer to this question in particular. What are we kind of missing from this picture, and kind of the general things of how ketamine works, and what is missing from the current way we conduct studies and investigate these matters. And in my opinion, that thing is sleep. And if you think about this timeline here, from the ketamine treatment to the maximum responses, what happens in between? There's actually quite a long period we spent completely sleeping. So within that 24 hours, we actually go to sleep. And I think that is crucially important for ketamine's effects to ultimately take place and perhaps be consolidated and sustained. And unfortunately, this hasn't been really getting that much attention in the field. So just a few words, I'll try to explain why I think sleep may be involved and how it's been also influential for the way I've been thinking about things. So if you think about any regular day of our lives, we wake up in the morning, we maybe go to work, study, we are active, we learn, we exercise, we be in social contact. This is pretty much how it looks at the moment due to the COVID situation, but more or less these are things that can fill up our daily lives. And essentially, after all these activities, I think many of us feel sleepy, and then ultimately all of us go to sleep at some point. And this essentially forms a cycle that we then go throughout most of the days in our lives, from birth to kind of elderly life. And if you think about this in terms of the synaptic homeostasis hypothesis, which is a kind of a theoretical framework proposed by Giulio Tononi and Chiara Cirelli, suggesting that the role of sleep is essentially to enable the brain to go to true plasticity. Sleep is the price the brain pays for plasticity, as they coined it. So the idea here is that throughout our waking time, the brain and its synapses essentially gain synaptic strength. So by learning, by interacting, by activating these various networks and neurons and synapses, they are gaining strength throughout the day. And then when we go to sleep, and into particularly to deep sleep, where there is an increase in the slow wave activity, and this slow wave activity is, by the way, proportional to the preceding levels of activity during the preceding day. That is a well-known fact. It's thought that then during this sleep period, the potentiation is then kind of renormalized or downscaled, and that we need this kind of a process to maintain our brain in a functional state, because we cannot just kind of constantly potentiate our synapses without ever kind of setting the system back. So that's one of the key ideas. Now, how does that then revolve to ketamine and its effects? Is essentially through many studies already done several decades ago in rodents, where they demonstrated that ketamine given to animals or also other NMDA receptor antagonists, that those actually increase the emergence of slow waves during the subsequent night or subsequent sleep period in rodents in particular. But also there's been a recent clinical study where ketamine infusion to depressed patients increased the slow wave amount of slow wave sleep during the subsequent night, and that appeared to correlate with the efficacy or the antidepressant effects of ketamine. And to me, that is kind of pointing towards the direction that this slow wave phenomenon is important for ketamine's actions. And indeed, this similar phenomenon where activity or kind of increased excitation results in the increase of the slow wave phenomenon is also present with many of the other putative or kind of also clinically used antidepressant treatments, such as, for example, theta burst stimulation or RTMS in general. And sleep deprivation, of course, causes increase in slow waves during recovery sleep. And, for example, ECT clearly increases the emergence of slow wave activity. So it's kind of suggesting to me, at least, that there is a common kind of homeostatic phenomenon that these drugs and treatments are somehow affecting. And many of these things here are more properly discussed in this recent review by our group. Now, we also recently proposed the hypothesis of encoding consolidation and renormalization in depression together with my PhD supervisor, Dr. Tommi Rantamäki from the University of Helsinki. And here we kind of go through how ketamine may act through mechanisms of synaptic homeostasis and sleep to elicit its effects on depression. And I'll just give a brief overview of what kind of what this figure is all about. So basically what we propose that in depression, here displayed as these abstract red and blue networks, we propose that there are networks which are hyperactive, for example, those such as the subcanal anterior cingulate or parts of maybe the default mode network. And those particular networks and circuits which are hyperactivated, for example, through depressive rumination or this kind of constant pondering of negative thought patterns, can then be potentiated, for example. Then other networks, which are kind of part of more balanced cognitive processing, might actually decrease in activity. And here's just the idea that, use it or lose it, and whatever you use and repeat, you also strengthen. So basically the idea is that in depression, networks related to the depressive processing are upregulated, and maybe other networks might actually be downregulated in terms of the kind of overall synaptic strength. Now, we think this is kind of could be a part of the process leading to depression. And what ketamine could be doing is that it's actually activating those cortical areas, cortical networks, and increasing their synaptic strength already quite rapidly and acutely, thus kind of making these networks more kind of eligible to compete with those hyperactive depressogenic networks. And what could then take place during the sleep of these a couple of subsequent nights is that this process of synaptic renormalization could then result in the modulation of these networks in a way where perhaps the balance is then again achieved, or at least the network balance is adjusted towards a state where the depressogenic processing is not the dominant type of activity, and thus that would then result in the amelioration of the depressive symptoms. Now, also one of the ideas is that since the ketamine symptoms eventually come back, that could take actually place during subsequent sleep periods during the following week or two, when if there's kind of a predisposition for some of the networks to go into this state, which then is associated with depressive-like activity, then that could then happen again during subsequent nights, unless there is something done, perhaps with the means of psychotherapy or kind of rehabilitation or something that then kind of maintains the effect that ketamine has started in the brain and in those networks and synapses. But I won't be going into any more detail here, so you can read this work if you're interested in this, and it's quite a big piece of our hypothesis. So make sure you have enough time to go through it in detail, and send me questions if there are any related to that. And with that being said, I would just like to end the presentation. Here you can find me online, and here are some of my funding agencies, the Sigrid Juselius Foundation and Orion Research Foundation, which I kindly thank for supporting my postdoctoral work. Of course, my colleagues at Weill Cornell Medicine and at my previous lab at the University of Helsinki, and collaborators at Art Lab Productions are thanked. Here are a few of the papers I mentioned in this short presentation that might be interesting to those who want to kind of dig a little bit deeper. But with all that being said, I thank you for your attention, and let's see if we have time for the questions.
Thank you, Samuel. Really beautiful visualizations.
All right.
So there's a question in the Q&A. I'm going to read it here. I remember that magnesium ions are the natural blocker for the ion channel in NMDA receptors. Have there been any research with magnesium and ketamine co-administered? If yes, what have the results been? And if no, what would you think could happen?
To be honest, I cannot remember any studies that would have concentrated on looking at whether magnesium combined with ketamine is really beneficial for the antidepressant effects. What I do remember is that there are several kind of exploratory studies investigating magnesium as one of the mechanisms of or how magnesium is related to ketamine's effects in preclinical studies. But I can't really remember that there would have been something kind of very important or crucial in those studies. Probably magnesium has some kind of a modulatory effect. What I left out from the kind of the basic receptor level stuff in the presentation was that magnesium is the kind of endogenous blocker of the NMDA receptors, which is then voltage dependently plunged out of the channel pore, and then allowing ketamine to enter and block the receptors. But then again, in more general sense, I'm not entirely sure it would be beneficial to enhance kind of ketamine's ability to to block the receptors if that's what the magnesium would be doing. It's a little bit unclear because magnesium is very voltage dependent in its blockade. And in that sense, it may be something closer to, for example, memantine, which hasn't been really found to produce antidepressant effects. But it's an interesting question and hasn't been really studied that much. It would be interesting at some point to see more studies about this.
Because ketamine has such a rapid acting effect on depression, and as you just introduced the mechanisms, neurobiological mechanisms of it, what can it tell us about depression in general and depression's origins?
Is obviously a kind of complicated question, and I, to some extent, I already discussed those ideas when talking about the NCORD hypothesis. And in general, what's particularly interesting about ketamine's effects is the very rapid amelioration of the depressive symptoms, and yet how they are sustained then during the following days and weeks. So for me, what ketamine's effects are telling in this context is that they are acutely changing how the brain is active, how some of the networks are active and connected already during or immediately after the ketamine infusion. And that probably doesn't mean that there is already at that point some kind of dramatic structural change taking place, but it's just that the ketamine is perhaps increasing cortical activation, and through that cortical activation, it is perhaps altering kind of protein synthesis, trafficking, causing like increases in synaptic strength, and that is enough to kind of alter how these networks then are active. And ultimately, their activity is what brings the amelioration of depressive symptoms and kind of the balancing of the networks out of such a state which supports, for example, depressive rumination. Now, the second thing is, of course, then what happens afterwards. And like I already briefly discussed, I think their processes, for example, protein synthesis during subsequent hours, and then ultimately whatever takes place during sleep, perhaps those processes of synaptic renormalization could be crucial for ketamine to then ultimately have its kind of more long-lasting effects on synaptic strengths, and through those synaptic strengths and their regulation also on the function and activity of these networks. So yeah, I think that's like a very short answer to a very complicated question, but something along those lines, at least to me, ketamine is telling about depression.
Great. How about what can this upregulating of the synaptic connectivities and then during slow waves downregulating it? Is it related to others, the mechanisms of other psychedelics as well? What do you think?
Yeah, that's something we've been already thinking about, and we briefly discussed also in the NCORD hypothesis. And particularly what I think is key here is that all of these putative rapid-acting treatments of depression, or kind of more experimental treatments, they all share the ability to more or less excite cortical networks and increase cortical activity. For example, you have the RTMS, or theta burst stimulation, which is just kind of stimulating a certain brain area. You have treatments like electroconvulsive therapy, which trigger more or less generalized seizures, which are kind of taking a lot of brain areas at the same time and activating them strongly. And then you have treatments like sleep deprivation, which also put the cortex and many other brain regions on kind of a hyper active state until the recovery sleep then comes. So I think the kind of the activation, cortical activation in general, is a shared feature of all these different treatments that are effective also clinically. And I think psychedelics could also fit into that category. So particularly those binding to 5-HT2A receptors could be causing something similar, since those receptors are also excitatory in nature. And they have a particular expression pattern in areas that could be particularly interesting and useful for trying to change patterns of connectivity and activity in regions which are implicated in depressive thought patterns and so on. So yes, I think these ideas could very well be also relevant for psychedelics, and to my knowledge, nobody has really yet studied sleep in the context of the antidepressant effects of psychedelic drugs at the moment. So that will be an interesting future avenue.
Okay, thank you for the great presentation. Unfortunately, our short time period is up, and we will continue in the panel discussion with Samuel. Thank you very much, Samuel.
Thank you.